Hey guys, so this video is over cell transport or membrane transport in a cell. It is really important that we understand how this works because your cell membrane is designed to keep certain things in. It's also designed to keep certain things out, but at the same time, we need to be able to move substances across the cell, like water and other molecules, because things are needed for the cell to function, as well as we need to get things out so that other cells can function and do their job. Okay, so this is all about cell transport. And this is covering section 3.3 in your textbook. Okay, I will be typing most of this. I'm still learning how to not write like a 5-year-old on this this pen tablet thing that I have. So I'll be writing a little bit later, but most of this will be typed for right now. Okay, so what we should have learned already on this is that the cell membrane is what we call selectively permeable. And what this means is that it lets certain things in and out, but not everything. Okay, it might allow water to come through, but it's not going to allow large molecules of glucose to come through. It's not going to allow molecules of different charges in and out. So we have to be able to control, again, what comes in and out of a cell, because it is what helps to keep your cells healthy and then in turn keeps you healthy. So if it's selectively permeable, it's going to let some things through, but not others. And what we'll learn as through the course of this little mini lecture is that some things that your cell membrane won't let through still have to come through. So we have what we call carrier proteins that are embedded in the membrane, and they help to get some of the larger things through that can't usually get in on their own. Now, there's a couple different types of cell transport. We're going to go through each one of those here in just a minute. Okay, the first one of those we call filtration. And that is exactly what it sounds like. Is your blood or tissue solutions going through a filter. Okay, some things stay in, rest of it goes through. Okay, so it's essentially like when you make coffee in the morning. Okay, you put your coffee filter in and you fill it with grounds, and you put water in the machine. The water goes in through the grounds, and that filter lets the water go through along with some of the molecules from the coffee, but it keeps most of the coffee grounds, the big chunks, from getting into the coffee you drink in the morning. Okay, so it's kind of the same thing with your blood. Okay, this is going to happen because of the physical forces that are pushing fluid through. Okay, driven by an example of this in your body is going to be the blood through capillaries. So at the front end of a capillary bed, and we'll learn about this when we get to the circulatory system and circulation. At the front end of a capillary bed, the pressure is so high that it pushes a lot of things out of your blood vessels through the spaces between the cells. And then at the end of the capillary bed, the pressure outside of the veins is going to be higher, so it pushes things back in. So it helps to push a lot of the ions and some of the water and things out of the blood, and then the waste and things come back into your capillary to get back to your heart and for things to leave your body. Okay, so filtration is one way that happens, and usually, so blood, capillaries. So the components of your blood are pushed through the capillaries that are there. I want that to be number 2. Okay, well that's supposed to be number 2. I don't know how to fix it. So the second type of transport we have in our cells, what we call simple diffusion. And simple diffusion is the movement of molecules from an area of what we call high concentration to low concentration to equal. And I am writing molecules in all caps because this simple diffusion does not really include water. Okay, and that's kind of important. So essentially what we have, and here's where I'm going to try to write as neat as possible. Okay, when we have simple diffusion, we're moving from an area of high concentration. So if you're not familiar with this shorthand, this two brackets closed or closed brackets around each other, that is a shorthand for concentration. So all molecules will move from an area of high concentration to low concentration until, oh my gosh, until equal. Okay, so if you think of it this way, this high to low until equal is the natural movement of molecules all the time. Okay, molecules move around a lot and they're bumping into each other, and they want to have enough space so they're not bumping into anything. So what's going to happen is that there's a high concentration of molecules on one side of a membrane. It's going to, because of the movement of molecules and the bouncing around and vibrating of each other off of each other, they're going to move from the high concentration to low concentration. Okay, and you can think of it as if you were in a room and somebody stuffed 200 people in that room. Your natural tendency, I would assume, is to get out of that room because there's too many people around. Okay, that is moving from an area of high concentration to low concentration. Okay, where it levels off is when that concentration is equal. Okay, so let's say I've got a membrane here, and let's say I have a concentration on this side of 2 milliosmolar per liter of, I don't know, pick any random molecule. You can pick calcium. Okay, and on this side, we're going to have 100 milliosmolar per liter of calcium. Okay, again, the natural tendency is for those molecules to move from high concentration to low concentration until the concentrations are equal on either side of that membrane. Okay, so that is simple diffusion. Now, there's a couple things that will essentially affect the rate at which diffusion occurs. Okay, and they are as follows. So the first one is temperature. Okay, at a higher temperature, your molecules are going to move more rapidly than they will at a colder temperature. So if you heat up the liquid that is here, okay, this movement is going to happen much quicker than if it were cold. Okay, so it's faster and warmer. Okay. Then temperature. We also have the molecular weight of the molecule. Okay, so what's the molecular weight of whatever molecule we're moving, or we're moving from one side. Okay, so if we're moving carbon, okay, that's going to move a lot faster than calcium because calcium has a higher molecular weight. Okay, so higher weight is going to equal slower movement. So the molecules that have a higher weight are going to move across that membrane much slower than if it were this up. Oops. Than if it were a smaller molecular weight. Okay. Okay. Now, what also is going to affect the rate of the diffusion across the membrane is going to be the steepness of the concentration gradient. Okay. So, for example, if we're talking about this situation over here, we have 100 milliosmolar on one side and 2 on the other, and we were going to compare that to, let's say, put another one right here. Say we have 2 again on this side, but now we have 10,000 on this side. The movement in this situation is going to happen much faster than at this situation. Okay, so the higher the gradient, the faster it's going to move. Oh goodness. All right. What else is also going to affect the rate? It's going to be the amount of surface area. of the membrane. So if you have more area in which you can move these molecules across the membrane, it's going to happen much faster. Okay, so for example, the cells that line your intestines, they have what we call a microvilla, or the margin of the cells is going to look like this. Okay, so it's going to have essentially little finger-like projections, as opposed to another cell that might just be straight across. The surface area in this cell is much higher, so this cell is going to move molecules at a much more efficient rate than, say, this flat cell here. Okay, so more surface area, more movement, and then also the permeability of that membrane. Okay, so if that membrane is much more permeable to that substance, then it's going to move that substance much faster. Okay, so all five of these are going to be situations and characteristics that are going to affect the rate of diffusion across a membrane. Okay, so again, remember, simple diffusion is going to be your movement of molecules. Okay, that's really important to remember. We're talking about molecules here, because when we get into the next one, we're going to talk about water. Okay, so the next type of cell transport, okay, is osmosis. No, I want 3. Okay. Osmosis is the movement of water, okay, and water only. When we're talking about anything but water, you want to use the term diffusion. When we're talking about water, we use osmosis, okay? If you remember from your readings about the cell membrane, you know that your cell membrane is a lipid bilayer, okay, which looks a whole lot neater, but similar to this right here. We've got your hydrophilic heads, or water-loving heads, facing outward, and then you have your hydrophobic, or water-hating, water-fearing tails facing inward, so that the heads, the sides that like water, are in contact with the water inside and outside of a cell. Right? Water can move, has the ability to move through this membrane freely. Okay, but water is one of those things that needs to be able to move quickly. Okay, so a lot of the water movement in a cell is going to be happening through a protein called an aquaporin. Okay. This is a protein embedded in the cell membrane, and it's going to move water at a much greater rate than if water were to just move through that cell membrane all by itself. Okay. Now, when water moves, okay, it actually is going to move essentially in the opposite direction that we talked about in diffusion. Okay, so water is going to move from, we call it a high solute concentration. Okay, and the solute is going to be the molecules that are dissolved in the water. Excuse me. So it will move from, oh, I'm sorry, I got that backwards. It will move from low solute concentration, my bad, sorry, to an area of high solute concentration. Again, this will happen until that concentration is equal, okay? And you can think of it this way: where there is low volume of solute, there is a high amount of water. Okay, and where there is high solute, there is low water. Okay, so water does move from an area of high water concentration to an area of low water concentration, right? But it's going to move essentially opposite of those the molecules in the solution. So let's think of it this way. So let's say I have my membrane here. Okay, on this side, let me work in some of my colors here. Okay, the blue dots are going to be water. Let me add a bunch of water dots in here. Okay, full of water molecules. Okay, this side we're not going to put as many. Okay, so we have a high water concentration over here and a low water concentration over here. At the same time, we're going to have a lower concentration of the molecule. We can, let's say, calcium, for example. Okay, lots of water, a little bit of calcium. And on this side, maybe we have a lot of calcium. Okay, or you can think of it as salt. Okay, so lots of salt on this side of the membrane where I'm drawing right now, and not a lot of salt on the other side. So this water on this side is super salty, and the water on the other side, not nearly as salty. Okay, let's also say that this membrane right here is not permeable to salt. So the salt, the green dots here will not be able to move. So the only thing that can move is the water, okay? And it will. So the water is trying to, again, remember the nature's tendency is to keep things equal all the time, okay? And because we can't move the solute concentration, we can't move the salt from one side to another, we've got to move the water. So the water is going to move from an area of low solute concentration and high water concentration to the other side over here, from the what they call the watery side to the not watery side. Okay, so water will move essentially in the opposite direction. Now, if we pretended that this membrane was permeable to salt, okay, to the salt molecules that are here, the salt itself, let me go back to green here, the salt will move from an area of high salt concentration to low salt concentration, okay? But the water and the salt will move in opposite directions of each other. Hopefully that makes sense, right? And this will stop once the saltiness of the water becomes the same on either side of this membrane. Now, there's ways that we can describe this the saltiness or the concentrations on either side of a membrane. Okay, we can go just a couple different ways. So let me set this table up here, make it look a little bit pretty. So we can have a couple different words we can use. We can describe a solution as hypotonic. Okay. We can describe it as isotonic. Oops. And then we can describe that as hypertonic. Let me make all these look the same here. Okay. Now, I'm going to start in the middle with the isotonic. Okay. Iso is a term that means the same. Okay. And essentially what this means is that the concentration of the solute is going to be the same on both sides of the membrane. Type 2 either. Okay, and it will essentially look like this. So for each of these, I'm going to draw a beaker of water, again, as best as I can. Not trying to look like a kindergartner. Okay, and you're going to have a beaker full of water. And in that water is one single red blood cell. Okay, each one of these red blood cells, and it's going to be the same through all of them, is going to have a concentration of 0.9% NaCl, because that's what your red blood cells are in your body. So you can imagine this as a red blood cell sitting in water. Okay. In an isotonic solution, the concentration of salt outside of the cell is going to equal that of the concentration inside the cell. So in this situation, we're going to also have 0.9% Na, looks like a W. Let's fix that. NaCl on the outside, okay? And that doesn't mean there's not going to be any water movement, okay? There's always going to be water moving, but what this means is that the amount of water moving out of the cell is going to equal the water moving into the cell, okay? So the size of the cell is not going to change at all, okay? So the red blood cell in this particular solution, so RBC is red blood cell, will stay the same, okay? It's not going to change size, not going to change volume, okay? That is isotonic, it means the same. All right, so let's move over here to the hypotonic, okay? In a hypotonic solution, okay, the solution is going to have a lower concentration of solute than the cell itself, okay? So your concentration of solute is higher inside of the cell. Okay? So what that's going to look like is this. So again, you've got your same beaker. With the water, with your red blood cell inside. Okay? Now, inside your red blood cell always is 0.9% NaCl. There we go. Okay. If the solute concentration is higher inside the cell, then we can say that we are in a solution now. Let's say it's 0.01%. Okay. And remember, the natural tendency of how nature works is it wants concentrations to be equal on both sides. Okay. In this case, salt cannot move through the membrane by itself. Okay. So in order to make these concentrations equal, your water is going to move from the watery side into the not watery side. So you're going to have a net movement of water going into the cell. Okay, you're going to have lots of water going into the cell. You'll have some that comes out, but it's not very much. For the most part, it's going to go in. Okay, and if you think about what might happen to the cell, okay, you hopefully will understand that this cell is going to swell. Okay, and eventually, if it does not get this under control, it will go through a process we call lysing. Okay, and essentially what that means is it's going to burst, it's going to break, it's going to kill that red blood cell. Okay, so that's in a hypotonic solution. Okay, more concentration inside of the cell. Excuse me. The very opposite situation is a hypertonic solution. So in a hypertonic solution, the concentration of solute is higher outside. So let me kind of show you what this is going to look like. Oops, I don't want that. I want it to be black. All right, so again, same beaker, a little bit bigger, I guess. Same water, same red blood cell, with the same concentration of salt, so 0.9%. Okay, now, but this time, we are going to have, let's say the salt concentration on the outside needs to be higher. So let's say we're going to put it at 5% NaCl. Okay, what this is going to do, because the solute concentration is higher outside the cell and nature wants everything to be equal, this is going to cause the water to rush out of the cell because it was trying to make the water less salty. Okay, again, there's going to be some water that moves in, but it's not going to be very much. For the most part, it's all leaving. Okay, and what this is going to do to the cell is it's going to cause the cell to shrink. Okay, or what we call crenate. Okay, it's going to shrivel up. It's going to look really weird. It's not going to work like it's supposed to. Okay, this would be like if you went to the hospital and they gave you an IV bag of straight water, deionized water, no ions in it, straight up water. Okay, this is going to cause your cells to swell and lyse, and you will probably die from it unless they fix it. Okay, this would be like if you went to the doctor and or you're swimming outside the beach and you swallowed a bunch of water. Okay, this is going to cause the cells around where the water is to shrink and crenate, which is what makes you sick when you drink, and you can die from drinking too much seawater. Okay, so if you have questions on this, let me know and I'll try to explain it a little bit better. So that is osmosis, right? Filtration, diffusion, filtration, simple diffusion, osmosis. This can happen in a normal living cell. This can also happen across artificial membranes, so dialysis tubings, things like that. Okay, now we're going to get into where we're talking about things that have to happen in a cell, and the molecules that are moving need a little bit of help. Okay, and there are two types of movement where the molecule is going to need a little bit of help. Okay, one of those is what we call carrier-mediated transport. Okay. And essentially what this means is that there are carrier proteins in the cell membrane that are going to assist in the movement of molecules, including water. Okay, so the aquaporins that help the water move in and out of the cell very quickly, that is considered essentially a carrier-mediated type of transport. But because it uses no energy and it works a lot, we cover that with osmosis. So in a carrier-mediated system, okay, it is very specific, okay, which means it only carries certain things, okay? So there's a transport protein for glucose in your cell. It is not going to also carry salt, for example. Okay, the one that is the sodium glucose transport, it is not going to carry sodium or potassium. Those two are going to go to their own pump, right? So it's very specific, and there are three types. Okay? Three types of transport, of carrier-mediated transport. Okay, one of those types is called a uniport. Okay, and I'm putting these in all caps just for emphasis, so you don't have to write them down that way. In a uniport, it moves one type of molecule. Okay, so an example of this in your body is going to be the carrier molecule that moves glucose. Okay, there's a molecule in the cell membrane, protein embedded in the cell membrane, that all it does is it moves glucose. Okay, we also have what we call a symport. Okay, this is going to move two types of molecules. In the same direction. Okay, so again, an example of this in your body is what we call the sodium glucose transporter. Okay, it's abbreviated the SGLT, sodium glucose transporter. Okay, so what it does is it moves sodium and glucose at the same time in the same direction. Okay, so that's a symport. And then you also have what we call an antiport. Okay, and what this is going to do is going to move two types of molecules in different directions. Okay, so the sodium, my letters here, oops, potassium pump. Okay, the sodium potassium pump is one of the most energetically expensive pumps in your body. I think it's somewhere like a quarter of all the energy that you consume every day is used to power these pumps in your cells. It helps with the conduction of neural activity across your neurons, things like that. So it's really important. But what happens is at the same time it's bringing, for example, sodium out of the cell, it is bringing potassium into the cell, and it's happening at the same time, but the sodium and the potassium are moving in different directions. So we have Uniports, one molecule. Symport and antiport both move two. Symport, think same. Symport, same. Anti, opposite, moves them in opposite directions. Now, we have those three types of carriers, or the three types of carrier-mediated transport. Okay, these three types, you find them in all of what we're going to talk about here in a second, but there are three methods, essentially, for movement of these molecules in a cell. The first one is what we call facilitated diffusion. Okay. In facilitated diffusion, okay, it's very similar to simple diffusion, where molecules are moving from high to low till equal. Okay. The difference is we need to use a carrier protein, a carrier protein for movement, because those molecules usually are large. They're opposite charged from the membrane itself, so they just need a little bit of help. Okay. So facilitated diffusion, again, it's movement from high to low until equal, but this time we're using a protein to help. In simple diffusion, you do not need a protein. Things are moving just through the membrane on their own. Okay, facilitated diffusion, they're large molecules that can't get through the membrane by itself. Glucose, for example. Glucose does not move through the cell membrane by itself. It has to have a carrier protein for facilitated diffusion. It will go from high concentration to low concentration until it's equal. Another cool thing about facilitated diffusion is it uses no ATP. Okay, when molecules are moving across down, we call down their gradient from high concentration to low concentration, we do not use ATP for that. Okay, that just follows a natural tendency of molecules in their movement from high concentration to low until equal. Okay, when we use ATP, we call that active transport. Okay, in active transport, we use ATP. Okay, now with hang on a second. Okay. With active transport, like I said, we are using ATP. Oops. There are two types of active transport. There's primary active, okay, and there's secondary active, and we'll talk about that in just a second. So in primary active transport, okay, you've got, we use ATP, okay, so because we're using ATP, we are actually going against the concentration gradient. What that means is we're moving molecules from an area of low concentration to high concentration. Okay, so we have to use the energy because we're going opposite of what is natural tendency of the molecules. Okay, so that is active transport. Again, we're using a carrier protein for that using ATP. Okay, in what we call secondary. Okay, again, we're moving things opposite of their direction. Okay, but what's different now is we're not actually using ATP for this. Okay, what happens is this is going to be a pump that is essentially relying on the gradient created by the active transport pump. Okay, so the primary one is moving things from low concentration, or from low concentration to high concentration. So moving things into a cell that wouldn't normally come into the cell at that particular time. The secondary active transport is using the gradient created by the primary active transport to move things from high concentration to low concentration until equal. Okay, so the secondary active one is not using the ATP to operate itself, but it essentially is kind of piggybacking off of the primary active transport carrier protein. So it does not directly use the ATP. Okay, so again, secondary does not directly use ATP itself, but operates off of the gradient created by the primary active transport. The primary active transport does use ATP. Okay, so an example of a secondary active transport pump in the body, and you'll learn more about this as we go through, is what we call that sodium glucose transport. So the sodium glucose transport, okay, this is going to rely on the sodium gradient that is created by the sodium potassium pump. Okay, and what this is doing is it's pumping sodium into the cell. Okay, that's creating a high concentration of sodium inside of the cell, which then allows this the SGLT pump to move sodium and glucose out of the cell. Okay, so again, it's relying on the gradient that was created by the primary active transport pump. Okay, so those are your carrier-mediated transport. The last type of transport that we're going to talk about is going to be what we call vesicular transport. Okay, so vesicles are essentially small bubbles that are created inside of a cell when things need to be moved in and out of a cell. Okay, so maybe your body or your cells making a hormone, and it is going to be created in the Golgi apparatus, the smoothie yard, then the Golgi apparatus, and then after it leaves the Golgi apparatus, it's going to need to be excreted from the cell. Okay, so the vesicle that comes off the Golgi that is carrying that hormone is going to fuse with the cell membrane and then push out its contents. Okay, that's one type of vesicular transport. Excuse me. Oh, I spelled that wrong. That was weird. Vesicular transport. There we go. That looks better. Okay. So there are two types of vesicular transport. Okay, and they are called endocytosis. Okay, and then the other one is going to be called exocytosis. Okay, so usually words that are ends in SIS is a process. Cyto means cell, endo means in. Okay, so in endocytosis, we are, it's a process by which we bring things into the cell. Endo means into. So we are bringing things into the cell. Okay. In exocytosis, this exo, I want you to think of exit. Okay. So in exocytosis, we are taking things out. Okay. So endocytosis brings in, exocytosis is letting things out. Now, under endocytosis, there are two processes that can happen, actually three processes that can happen in your body, if I could talk correctly. All right, the first of those is what we call phagocytosis. It essentially means cell eating. And it is the process of bringing large things into a cell. Okay, amoebas do this when they eat. Your I had a brain fart. Your but white blood cells in your blood, they will engulf objects into their bodies to kill them in order to kill bacteria and viruses. And there's this really cool video that I wanted to show you here. So this is going to be a neutrophil. That was the word I couldn't think of a second ago. It's a type of white blood cell in your body, and it's going to be swimming around a bunch of red blood cells. And what happens is, what it's doing here is it's chasing this little piece of bacteria right here because it's not supposed to be in the blood. It's trying to get rid of it. Okay, and what you'll notice here pretty soon, it's going to actually engulf that protein or that bacteria into the cell. It's going to create a vacuole, and that vacuole is eventually going to, so there it goes into your cell here. So now it's inside the cell. It's going to fuse with a lysosome, which essentially is like the garbage truck of the cell. And, oh, don't play. And it's going to kill whatever it brought into the cell. Okay, so that is going to be phagocytosis. You're bringing large things into the cell. Oops. Okay. Now, the other type is called pinocytosis. And essentially is cell drinking. So it is a way for the cell to bring in large amounts of water at once. And it does this essentially in the same way. Okay. So what happens is you have the cell wall, it's going to kind of, when it gets around the molecules it wants to bring in, it's going to invaginate or kind of cave in on itself. And eventually it's going to close itself off and it's going to create a vacuole inside. I remember this because pino sounds like peeing, and peeing is liquid. And so pinocytosis is bringing in a liquid, whereas phagocytosis is bringing in large molecules to eat, essentially. Now, the third type of this is called receptor-mediated endocytosis. There we go. Okay. And essentially what this is on your cell membrane, you're going to have these little, essentially these receptors. And once they receive the ligand, and the ligand essentially is the key that's going to unlock the process. Once the molecule that is the ligand gets into the key, gets into the lock, into that receptor, it's going to trigger the invagination of the cell wall in that area. Okay, and that's going to bring in a large amount of whatever that molecule is. Okay, there's a really good picture of this on page 95 in your book. Right. Once those extracellular molecules attach to the receptors, it's going to cause a little pit to form. That's that invagination of the cell wall. Okay, and eventually it's going to close itself off, and then that vesicle is now going to be inside of the cell. So whatever was in those receptors gets brought into the cell in that way. Okay, so that's receptor-mediated endocytosis. Okay, the last one is what we call this exocytosis here, and it is taking things out of the cell. And essentially what happens is the protein or fat or hormone or whatever it is made inside of the cell, right? It travels through that endomembrane system from the endoplasmic reticulum to the Golgi. When it leaves the Golgi, it comes off in its own little bubble or its own little vesicle. That vesicle will then fuse with the cell membrane, and then the contents of that membrane will spill out into the extracellular fluid, and it will then get to wherever it needs to go. Now, transcytosis is a kind of a smaller process that isn't mentioned very often, but this transcytosis essentially is you're taking material across the cell. So it's brought into the cell through endocytosis on one end of the cell, and it is brought out of the cell through exocytosis on the opposite end. So that's trans. Trans means across, right? Happens a lot in muscles, and it's kind of a pretty cool process. Now, any of these processes, you can look them up on YouTube, and there's a lot of cool videos that you can watch to kind of show you how these things work.